So first of all, this is by no means a unique problem to genomics. Plenty of other industries and research settings dealing with large amounts of data have faced issues and challenges like this and found ways of working around it. The second thing is that when you actually look at the methods and the software, the methods and the approaches for the analysis that are encoded in this software, what you find is that there's nothing really special going on here. So if you look inside a lot of genomics software or variant calling software, genomics analyses, I'm giving as an example here some of the equations that are behind a semantic variant caller. What you'll see is that there's a lot of pieces which are very similar to things that exist and are run at scale outside of the genomics environment. So I'd suggest that, for example, there's not at a mathematical or a fundamental structural level, there's not a whole lot of difference between, say, the pieces that go into a variant caller and the things that you might find inside a modern spam filter or some modern machine learning algorithm. There's some Bayesian stuff. There's some likelihood function stuff. There's some integration and sampling stuff. These are things that genomics analysis software do, but these are also things that software which operates at scale outside of genomics does. And what's important about that is that a lot of these tools, these integrations, these large sums, and bioinformaticians love to write down probabilistic models, which are just often large sums evaluated through something like expectation maximization or something similar. A lot of these particular algorithms or computations or formulae are well suited to running on top of next generation data platforms like Spark or Hadoop, running on substrates like Mesos or Yarn or other equivalent systems. The point being that a huge amount of the research that's gone on inside of the database and computer science communities over the last 30 years has been understanding how to take large computations like these and break them down and figure out how to execute them reliably at scale in a distributed environment. And that's really what things like Spark and Hadoop are about. So what I'd like to suggest is that it's time for us to start thinking a little bit about what a bioinformatics of the future would look like. And I'm going to point out, now we're in the sort of middle of this presentation, I'll point out a few of the tools that I've either used or that I've contributed to, and that I'll encourage some of you to take a look at or maybe even contribute to if you have the time and energy. I'll talk a little bit about some of these tools that I think are steps towards what a bioinformatics platform and software system of the future would look like. So the first one that I'll call out is something called the Common Workflow Language. There are a number of bioinformaticians who are trying to think carefully and openly. I'll return more to the open question at the end. But in an open setting about how to write down workflows like the ones that we've seen from these diagrams and papers. They're trying to make sure that there's a common and open ecosystem for languages that can manipulate these workflows or execute them, tools that can be embedded in these workflows, user interfaces that support the execution of workflows for authoring or capturing these workflows or putting them together inside of pipelines. So that's one thing I think that's really crucially important. I think we should all be thinking a little bit about how can we write software that makes it possible to join these systems and software components together in ways that aren't just simply running things in succession from a command line interface or something equivalent. The second thing, and one of the pieces of software that I've been thinking a lot about in the last year of my life, several years of my life, and I've been contributing to, I think is an attempt to rewrite some of the core routines and tools of genomics to run on top of platforms like Spark. So the primary project that I'd like to talk to many of you about today, or describe today, is a project called Atom. It's created at and hosted out of the AMP Lab, which is a research lab at Berkeley, University of California, Berkeley. You can see the URL for the data and the code and the project itself at the bottom of this slide. BD Genomics in this case stands for Big Data Genomics. The software itself is licensed under an Apache 2 license, and we've had a number of contributors from both research and commercial organizations. The reason why I think that that's important is that there are other software projects available, some of which try to make use of technologies like Spark or Hadoop. I'm not going to make any claim that Atom is any better at a technological level than some of those. I think it is in some cases, and it falls short in others. But one of the major things that software and projects like Atom have going for them is their openness and their licensing. They're built from the ground up to be used both by users in private industry and to receive contributions from people in private industry, as well as by people in public or research organizations. So I think that that's really a crucial point that I'll come back to at the very end.